In early June 2026, a major shift occurred in global biotechnology. The New York Times reported that a team of geneticists at Columbia University, led by Dr. Dieter Egli, successfully edited the DNA of human embryos with unprecedented pinpoint accuracy. Industry analysts immediately claimed that we are rapidly approaching an era where designer babies could become a reality within the next 5 years. To truly understand the scale of this event, let's break down what this technology is, how it works, and the latest test results. Part 1. What is it? Until now, geneticists relied heavily on the Nobel Prize-winning CRISPR-Cas9 system. However, CRISPR had a fatal flaw. It acted as molecular scissors, physically cutting through the double-stranded DNA. In response, embryonic cells often plunged into genetic chaos, dropping entire chromosomes and failing to develop. The Columbia University team utilized a fundamentally different approach called base editing. Instead of cutting the DNA strand, this method acts as a chemical eraser and pencil. It meticulously rewrites a single genetic letter within the DNA sequence without disrupting the overarching chromosome structure. Part 2. How it works. During the experiment, researchers focused on correcting specific severe pathologies. Using base editors, they precisely targeted and modified genes associated with dangerously high cholesterol levels and mutations responsible for fatal forms of anemia. The editing process was introduced during the fertilization stage, when the embryo consists of just a few cells. Because the method is so gentle, it bypassed the embryo's destructive DNA damage responses, allowing it to develop normally and produce healthy stem cell lines without the collateral damage that previously caused early embryonic arrest. Part 3. Test results in the 5-year outlook. The official findings demonstrated perfect accuracy in target editing without any genotoxic consequences. This safety profile is exactly why experts are projecting a 5-year timeline for clinical readiness. Science is not yet ready to engineer designer traits like eye color or enhanced intelligence, which remain heavily restricted and ethically condemned. However, within the next 5 years, base editing is highly likely to enter legal clinical trials inside IVF pipelines. This will allow parents carrying high-risk mutations to completely purge fatal inherited diseases from their future children's genetic code before embryonic implantation.